everyone, and welcome to our podcast. I am Narazura, and I will be kicking off today's session by introducing the exciting world of omics technologies, specifically how functional genomics and proteomics are reshaping the way we study and protect our environment. We all know environmental issues like pollution, climate change, and biodiversity loss are growing more complex. Traditional monitoring methods like testing water pH or measuring the chemical levels are useful, but they don't tell us what's really happening inside the living system. That's where omics technologies come in. Genomics allows us to study the DNA and gene expression, revealing what organisms are capable of doing in the environment. Proteomics, on the other hand, goes a step further by examining which proteins are actually being produced, telling us how organisms respond to stress at a molecular level. To show you why this matters, let's look at a real incident here in Malaysia, the Pasir Gudang chemical pollution case back in 2019. In 2019, illegal dumping of industrial chemicals like benzene and acrylonitrile into the Kim River caused over 6,000 people, and many of them school children, to fall ill. People were coughing, fainting, and also panicking. But more importantly, the incident exposed the gaps in our environmental monitoring systems. The air and the water were tested for chemical traces, but no one could confirm how organisms or humans were biologically affected. What if we had tools to detect early biological changes before severe symptoms appear? What if we could monitor recovery, not just clean up? That's exactly what omics technologies offer. So, in today's podcast, each of us will cover how different omics tools like metagenomics and proteomic biomarkers and eDNA-based biodiversity surveillance help us monitor the environmental stress, respond to pollution, and also improve the sustainability efforts. So next, speaker number 2, which is Isaac, will walk us through how functional genomics and metagenomics are being used to study the microbial communities and detect environmental threats at the gene level.
Hey everyone, I'm Isaac. We've all heard about the Pasir Gudang chemical pollution back in 2019. Thousands affected, especially the kids with symptoms like nausea and breathing difficulties. But what didn't get much attention was the environmental side of this story, especially how microbial life responded. So why focus on microbes? Because they're nature's first responders. When pollutants like benzene and acrylonitrile hit the soil, the river, or even the air, the microbes, they react immediately, even before we see damage in plants or animals. This is where metagenomics comes in. It allows us to extract and sequence all the DNA from the environmental sample, whether from soil or water. This gives us a very detailed look into what I think what microbes are there, and what's most important, what they're capable of doing. For example, in a site like Pasir Gudang, we'd look for genes that code for xenobiotic degradation. Basically, enzymes that they help break down pollutants. Species like Pseudomonas or Sphingeromas might thrive because these species, they carry genes for enzymes like monooxygenases and what's the other thing? Dioxygenases. These are the key tools for neutralizing toxins. But microbes don't just clean up, they also adapt in polluted environments. We often see shifts in microbial diversity. Some microbes disappear while others dominate, and sometimes they even share genes through horizontal gene transfer, literally passing pollution-fighting genes to help each other survive. Why is this important for us? I think it's important because even the chemicals vanish from human samples, as they did in Pasir Gudang. The microbial DNA keeps the memory. It acts like a living black box. It holds molecular evidence that something toxic happened. That's why microbes can also serve as bioindicators. It helps us detect pollution and track recovery. And the best part, we can use this data to guide bioremediation, either by enhancing native microbe activity or introducing pollutant degrading strains to clean up the site more effectively. So in my opinion, what I would like to say here is that basically microbial genomics, it helps us uncover invisible damage, they track ecosystem responses, they power new ways to clean up polluted environments in cases like Pasir Gudang. It offers insights that are crucial not just for understanding what went wrong, but for preparing smarter responses in the future, in my opinion. And yeah, that's it for my part. Thanks for listening. I'll now pass it to Nurin, who will talk more about genomics. Go ahead, Nurin.
Hi everyone, I'm Nurin. Picking up from Isaac Genomic Insight, I will now talk about how we use genomic tools to assess biodiversity and monitor ecosystem, especially in the aftermath of pollution events like Pasir Gudang. After a whole major incident like that, it's not just people who are affected. Entire ecosystem experience stress, species may disappear. Invasive one may take over and biodiversity can shift dramatically. But here's the challenge. Traditional monitoring methods like field survey can miss a lot, especially when species are hidden, seasonal, or live in hard-to-reach areas. This is where eDNA or environmental DNA become incredibly useful. It's basically DNA that organisms left behind in water, soil, or even air. And by collecting and analyzing this DNA, we can identify which species are or were present, including rare or elusive ones. For example, after Pasir Gudang, eDNA could be used to detect changes in species presence in nearby rivers, mangrove, or soils, giving us insight into biodiversity loss and recovery. But genomics doesn't just help us assess damage, it helps us predict. By building baseline genomic profile of ecosystem in industrial areas, we can detect early signs of ecological stress in the future. This is called genomic surveillance. Sudden changes in species DNA signal or gene function like stress or detoxification markers can act as early warning system. And when paired with data analytics or machine learning, genomic surveillance become a powerful predictive tool. We can even link gene activity with pollution levels to guide policy, zoning, and emergency responses. In short, while the Pasir Gudang case was seen as a health disaster, it's also a lesson in how genomics can help us monitor, protect, and restore its ecosystem. Genomic tools give the environment a voice, and it's one we can no longer afford to ignore. Thanks, and now I'll pass it to Sufyan to discuss the role of proteomics in environmental science.
Hi everyone, I'm Sufyan. So far, we've learned how genomics reveals the potential in microbial communities. Now, let's focus on proteomics, which shows us what's actually happening and how organisms respond in real time to environmental stress, especially pollution. Let's begin with pollution biomarkers. These are specific proteins that change when organisms are exposed to toxic substances. During the Pasir Gudang chemical disaster in 2019, thousands of people were affected. But when tested later, their blood and urine didn't show chemical traces. Why? Because many industrial toxins like benzene or acrylonitrile break down quickly in our body. That's where proteomics becomes important. Even when the chemicals are basically gone, protein expression changes remain. For example, studies show that exposure to benzene alters levels of proteins like ApoA1 and transthyretin, which affect lipid metabolism and immune function. These changes can be detected through proteomic tools like two-dimensional difference gel electrophoresis and MALDI. Other proteins like cytokines such as interleukin-6, tumor necrosis factor alpha, and interferon gamma are also involved. They indicate inflammation and stress responses, helping us detect early damage even when no symptoms are visible. Now, outside the human body, proteomics is just as powerful. In ecosystems, rivers, soils, marine life organisms express stress-related proteins in response to pollutants. For example, cytochrome P450 enzymes and heat shock proteins increase when fish or mussels are exposed to toxins. These serve as early indicators of environmental contamination. Heavy metals like cadmium and mercury trigger the expression of metallothioneins, proteins that bind metals inside cells. Measuring these can help us identify polluted sites before large-scale damage occurs. What's more interesting is how species adapt. Taxiona robusta, a marine organism that changes its protein expression under different salinity or temperature conditions. These physiological shifts help us understand how pollution impacts the survival, growth, and ecosystem balance. So in short, proteomics give us real-time molecular view of stress responses and environmental health. It complements genomics perfectly, where genomics identifies the blueprint and proteomics shows the action. And with that, I'll hand it over to Azura, who'll explain how we can use both genomics and proteomics to actually restore and remediate polluted environments like Pasir Gudang.
Thanks, Sufyan. So now that we have explored how genomics and proteomics help us understand what's happening at the molecular level during the environmental pollution. So now let's talk about how we can use these tools in environmental remediation and how this all comes together, especially in the cases like Pasir Gudang. Environmental remediation is basically about cleaning up the polluted environments and also restoring the ecosystems to a healthier state. Traditional methods rely on physical cleanup, chemical treatments, or natural recovery, but those alone are not always efficient, especially when dealing with invisible pollutants or complex ecosystems. That's where the functional genomics and proteomics come in. These technologies help us design the targeted and smarter remediation strategies. For instance, from the genomic side, if we know which microbes have genes for breaking down certain toxins like benzene or acrylamide, we can especially enhance their activity through biostimulation or bioaugmentation. We either stimulate native microbes with nutrients or introduce specialized strains to speed up pollutant degradation. In Pasir Gudang, microbes that carry genes for degrading volatile organic compounds could be encouraged to flourish in contaminated soil or river sediment, which helping to clean up naturally and sustainably. Genomic monitoring also helps us track how effective these strategies are over time. However, from the proteomic perspective, we can monitor protein level responses during the cleanup. For instance, if detoxification proteins like cytochrome P450 or metallothioneins begin to normalize in local organisms, it's a sign that the environment is recovering. This helps regulators and environmental terms fine-tune their approach instead of just relying on chemical testing, right? Even better, combining both omics approaches gives us a full picture. Genomics shows us the potential of organisms and what they can do. Proteomics tells us what's actually happening and how they are reacting to the stress or cleanup efforts. Together, they give us a powerful toolkit for both diagnosing and solving environmental problems. Now to wrap up, what we have seen today is that the functional genomics and proteomics are not just lab tools, but they are real-world technologies with the power to improve the environmental health, predict the ecological risks, and guide how we respond to disaster like Pasir Gudang. Whether it's identifying the resilient microbes, tracking protein strike signals, or shaping next-gen remediation strategies. Omics technologies are also helping us move forward a future where we can better protect both ecosystems and public health. And ultimately, the goal is not just clean up, it's about building resilience, making sure that when environmental crises happen, we have the science, tools, and knowledge to respond faster, smarter, and more sustainably. Thanks for listening to our podcast. We hope this sparked new awareness about how cutting edge science is transforming environmental conservation. Until next time!